Welcome to Flow Stars, candid conversations between Dr. Peter O'Toole and the big hitters of flow cytometry. Brought to you by Beckman Coulter at Bitesize Bio.
Hi, today on Flow Stars, I'm joined by Rui Gardner, flow cytometry core head at the Memorial Sloan Kettering Cancer Centre, and we discuss the pitfalls of undergraduate teaching.
I think that biology degrees or research-oriented degrees should essentially teach you how to think, to be critical, how to evaluate things in a very unbiased way. And I think that's pretty much the thing that we've lost.
The importance of conference happy hour.
This is just a typical example of how I used to drink in these conferences. You know, you would start with a glass as you see there, and then we would just start asking for pictures, right? And why not have a picture of sangria?
Why core facilities are such a good match for Rui.
It was a bit of everything that I always wanted. It was the science, the interaction with people, the non-pressure of having to publish. I remember thinking, I don't have to publish anymore, and a huge weight just left my shoulders.
And why leaving your comfort zone can be a really good thing.
Leaving your comfort zone is one of the most difficult things, but it's also one of the most gratifying things when you start becoming successful.
All in this episode of Flow Stars. Hi, I'm Peter O'Toole from the University of York, and today on the Flow Stars, I'm joined by Rui Gardner from the Sloan Kettering Institute over in New York. Rui, how are you today?
I'm doing very good. How are you?
I'm good. It's been a while since we met. I've got to say, you're looking rather dashing with the new
look.
And obviously, you even done your hair for this.
Well, you know, I realized that people didn't really take me seriously. So, you know, I looked, they kept saying, you look so young. And that actually is, I mean, in a way it's good. But on the other hand, when you really want respect, it's not that good. So I decided to grow a beard, and yeah, I've actually gained a lot of more respect from people now. So
yeah, now you look old.
Exactly. Old and wise, you know, you can trick people like that. I would encourage you to grow a beard.
Yeah, you want to make me look younger.
Well, maybe.
Anyway, so obviously you're not a native of New York. Where did you start with your undergraduate?
So my undergraduate, so it was all essentially done in Lisbon, in the Faculty of Sciences of the University of Lisbon. And I studied biochemistry, and it was a rough start because, you know, I dreamt. So my passion for science started, actually, I'm looking at the book that I read when I was 12 or 13 years old, and it was The Life of Pasteur, and I was really intrigued with the fact that, wow, this guy had ideas, and as a child he would try to find tubes and do experiments, and I just thought that was, you know, in his own house. And then as he grew up, he built his lab in his house, and I thought that was amazing. Like I can do the same. I can start asking all these questions that I don't have an answer for, and it really got me going with science. And you know, by the time I was 18, I was convinced that I really want to follow research. And I didn't know much about, you know, what would be the best degree for science and to become a scientist. And I really loved biochemistry, but I didn't really understand what biochemistry was at that time, because I thought biochemistry was more biology. What I know now is biology. So I actually think that I should have gone to biology rather than biochemistry, because biochemistry had all that part that I actually dislike, which is memorizing all the structures of the molecules, etc. And that's not really what I wanted to do. I wanted to follow, you know, I wanted to study biology. I was really intrigued with, photosynthesis, the respiratory chain, all these kind of things, which is biochemistry, right? But it was more maybe biochemical, you know, regulation, you know, how the system reaches homeostasis and things like that. Those for me were the intriguing questions. And so I started, when was that? 1991. That was when I started my degree. And as I said, it was a bit of a rough start because I thought, you know, I had this naive notion of science. I'm going to, you know, we're going to sit down, our teachers are going to give us problems, we're going to, you know, brainstorm, trying to figure out these questions. I thought I was going to work in a lab. But no, that was so naive. It was basically, I remember my first class, we were sitting down, hoping for an introduction and to feel really excited. The teacher came, the teacher didn't even look at us. He just turned around and he started writing on the board, and it was math. And he just started writing on the board, didn't say anything. And it was the whole class. We thought we were actually being, how do you say, what's the word when you're new and they do something to you?
An induction type thing.
Right, hazing, right? Something like that. And I thought it was a joke, but it wasn't. It wasn't a joke. It was just like that. Teachers didn't care. It wasn't about thinking. It wasn't about the thought process. It was all about just providing material. At the end of the year, you do, or at the end of the semester, you do an exam. And it wasn't even a continuous evaluation. It was just do an exam, whatever grade you have in that exam is the grade you have for your discipline, right? And so it was very disappointing. My first year, I just dedicated myself to the lab work. Really enjoyed the lab work. And then after a year, I thought, well, now I have two options. Either I continue, but now I have several disciplines that I have to work on and catch up, or I'll just say, okay, I missed this year. I actually did all the practicals, so now I only have to dedicate to the theoretical part. And so I decided to stay a year behind and just do the rest in a row. So it took me another 5 years. So total it was about 6 years. The fifth being actually working in the lab. And I can tell you about that, because I ended up not going to an actual wet lab. By my fourth year, I was already also convinced that I really loved applying math to biology. It was just such an exciting way of formalizing biology, because you think of biology as so complex, it's so hard to predict, right? And here was a tool where you could write things down and you could make predictions. And I thought that was really, really exciting.
So you went from not liking things being just written down and wants to be practically in the lab to then liking just writing things down and predicting what you might have done in the lab.
Right. So that's that's an interesting way of putting it. But what I didn't like was the memorizing part, right? Just memorizing for memorizing. What's the usefulness of it? Why am I memorizing this? I can do that later. Or if I need this, this material, I know how to read. I can go to the books and learn whatever I want. So I thought that was a waste of time to force us to learn things that we have no idea. Actually, we didn't know when we were going to apply it. And actually, we didn't know to what to apply it to, right? Especially math and the physics. You had all that material, or chemistry also. You had all that material that you didn't know when would you apply that. And you apply that when you start getting to more advanced biology and actually trying to answer questions, right? But that's the thing. If you have, if you're going to study, genomics, right? Then you study everything around genomics, right? How the DNA works, how genes work, how they're expressed, etc. But if you're going to be studying, I don't know, population dynamics, there's not much that you need to know about, DNA, right? I mean, it's always helpful in generic terms, but not at that extent, right? So we were learning a lot of material, which I thought, maybe one day it's going to be useful. But when you're young, and you're at the prime of your life, and you're asking, how do you say, you're asking questions that typically people, as they grow older, they might not have, the audacity to ask, right? When you're young, you challenge everything, right? You challenge absolutely everything. And as you grow, you start becoming more and more, you start having more and more prejudice about everything, right? So that's why it's very important. Actually, a small parenthesis here, I think that biology degrees or research oriented degrees should essentially teach you how to think, to be critical, how to evaluate things in a very unbiased way. And I think that's pretty much the thing that we've lost, well, or maybe never gained in many degrees. And I think that should be the way to go, rather than just learn so much material.
Do you not think that learning that material gave you the foundations to then go and ask the questions, because then you know what you can ask. You know it
Right.
exists, and you know where to go look for answers.
Right.
If we go back to our biochemistry textbooks, half of it is no longer relevant.
Right.
Things have
Exactly.
moved on, technology's moved on, answers, the science has developed quite rapidly. I started in 91 as well.
Right.
And I just sat down and just got on with it because I quite enjoyed the nightlife. So,
right.
And research, yeah, and actually I chose biochemistry without knowing what biochemistry was properly.
Right.
So it sounded like a good thing to do.
Very similar to me.
Yeah. But yeah, once you got in the research lab, and yeah, I remember, especially the chemists, some of the chemists, not all, just back to you, and there's a chalkboard.
Right.
There's a thought. It was chalk back then, and it was just one line down, 3 lines across. There's your carbon chain. C's here, oxygen's there, nitrogen's there. Next, arrow goes to this. Arrow goes to this, and it was just throughout. But so I thought that no context whatsoever.
Right.
Once the context came into place, I had that ammunition. I think biochemistry is going to be one of the best degrees out there, because if you do anything with a cell, it's all driven by biochemistry.
Right.
And when biology doesn't turn out the way you expect it to, it's because the biochemistry is different.
Right. I actually viewed biochemistry as a more exact science than biology, right? So it was kind of bringing the math, and the physics and the chemistry into biology. And actually, my mentor, the person that actually started the degree in biochemistry, I believe 10 years before I started. So it's probably in 81 that it started there in Lisbon. And this guy had been actually Hans Krebs' PhD student. And so he was pretty driven, excited. He was all about brainstorming, the thought process. He would used to go to the labs and make sure that people were doing things correctly, right? He didn't care about the whether they had knowledge or not. It was more best practices. It was all about best practices, and that really and that's coming to your point, I think those are actually the more important things that stuck with me. I agree with the fact that you do build ammunition when you're reading all these things, but it's a very inefficient way of doing things. That's my way of thinking it. So this guy that started actually had the same name as I, Rui, also. And for him, he used to say biochemistry is maths, chemistry, and physics. And as long as you have this great foundation, then you can learn anything in biology. Everything is just details. And I really that sunk in, because that's why I thought it was such a waste of time learning all this material. Give me questions, or let me answer these questions, and I'll have to find that material. I'll learn all the bases, right? And so I think the subjects should be taught based on questions, right? That force people to go there. Maybe I'll learn a different part of that foundation. Somebody else will learn a different part of that foundation. But it's part of finding the information that you need as a biologist, as a researcher later on. Because later on, when you're asking questions in the lab, you're not going to go, okay, so where has this been answered, right? Where are the books? The thing is, if you lack those foundations, you'll go and find them, right? You'll go and search.
I think you're right to a degree that there are those that will come out with the top degree that are very good at regurgitating information, reciting information. And there are those that are maybe come out with a lesser degree, but actually are very good at applying and finding solutions. And actually, from I guess from an academic research perspective, it's the people who can actually take what they know and apply it, find the information and apply it that are most important.
Right.
And there is different. I've got you obviously, for those who are listening, I think Rui might be sitting in your lounge, dining room, office.
No, in my living room.
In your living room. And you seem to have quite a bottle collection just behind you.
Yes, I do.
And is that all alcoholic drinks?
Yes, these are all alcoholic drinks. This is my well, actually, this is what I have for lunch or for dinner. So I always have them with me in my office here.
And so that is quite a collection. And you sent me, so I looked at, I hadn't noticed. And then I looked down and thought, oh my goodness, that there's a lot of spirits down there. It's not a haunted, right. These are alcoholic spirits. And actually, you sent me some. Oh, go on.
There you go.
That's quite impressive. And I love the way you put your children around your drinks.
Exactly. I put all the important things together.
Right to start with. But you sent me some pictures as well. And now I understand why you sent me this picture, because this is a picture of you drinking directly out of a pitcher of sangria.
Right, of sangria. So I forget exactly where this was, but all the pictures I sent you were pictures either, where I used to work or of conferences where I went. So I believe this must have been either a conference in Lisbon or maybe in Valencia, where, this is just a typical example of how I used to drink in these conferences. You would start with a glass, as you see there, and then we would just start asking for pictures, right? And why not have a picture of sangria directly? I mean, you can have a picture of beer, right? So, so that's the way I like to see myself in these conferences. I mean, they're extremely important. The science, the technology is extremely important, but the networking part for me is the most important of all. This is one of the things I love of flow cytometry is how we are able to. So when I started in flow, I had come as I was a scientist, and I used to go to scientific conferences. And I remember, again, with this enthusiasm of, I wanted to, there were people that were doing work very similar to mine, and I was the only one that I knew that was doing what I was doing. And so you would find somebody else asking the same questions and addressing them in a slightly different way. And that for me was so exciting. Jesus, there's somebody else. First, it's a validation that maybe what I'm doing is also relevant, right? If somebody else is also working on that. So, and then the fact that they also knew and had thought about it, and I could discuss with them. So I remember reaching out to these people, like in a poster session or something like that. And then, their first reaction, and this happened a few times, their first reaction was, uh-oh, there's somebody that's doing the same thing as I do, right? So they're going to take my work, publish it, and then I'm done. And I didn't think like that. I have to say, I'll just say, it's not a naive. I thought that maybe I was being too naive, but it's not. A lot of I've had the fortunate. I've been fortunate to actually attend several lectures of Nobel Prize winners, and for me, the thing that I hear that's common to all of these people is they always say the best thing, if you want to reach, this level, the best thing to do is share everything you do. Like, don't keep it to yourself. And I thought that was very strange, because what I'm learning as a young scientist is actually the opposite. I try to share my research and they just close themselves, even worse. They might take my ideas and then use them. And I thought that was very strange. And so there was a lot of, how do I say, the atmosphere it was very competitive. And I remember the first conference that I went, which, by the way, I don't know if you were there. It was in the German Society meeting. I think it was 2007. I think maybe we met in Flow Cytometry UK, also maybe 2007 or 2008. And I remember, these conferences were local conferences, right? And the people there were just amazing. Everybody was relaxed. There was no competition between, hey, you're doing that, then you're going to steal that away from me and publish it. No, it was just a bunch of people. Well, who did I meet? Andy Riddell, Alexis already knew, but, Ray Higgs, all these folks from the UK. Again, I don't remember if you were there, that one, or for Flow Cytometry UK that I met you, but, Rachel was probably there, Rachel Walker, all these people. And it was just a lot of fun. We would go to bars. I think Alfonso was also there. Alfonso Blanco, and it was just fun. It was just and I could ask all the questions. I had just started maybe 6 months in flow. I had no idea what flow cytometry was when I started. Actually, the person that was
Why did you choose flow cytometry? You didn't know what it was when it started.
Right. I didn't choose flow cytometry. So yeah, I can tell you how it happened. So I was doing a postdoc, computational biology. I was actually in a theoretical immunology group, and I was studying. Basically, I was trying to understand the interaction between T cells, APCs, and so effector T cells, regulatory T cells, and how the regulatory T cells were able to regulate the effector T cells. Did they bind to the APCs, and then the APC would now recognize the effector T cell, or was it an interaction between the regulatory T cell and the effector T cell at the same time on the APC? So we were trying to figure out if, by modeling this. So I was basically programming, and it was graphical. I remember doing this with C, programming language. But it was graphical, right? So you had the balls were the cells. They were just floating randomly, and they would touch each other, and I would change the rules of the game, right? So in one scenario, for instance, the regulatory T cells wouldn't do anything to the effector T cells. They would only activate the APCs, right? And the other scenario, they would have to be both together with the APC to be able to have let's say an effector mechanism. And so I tried all these scenarios around. So that was my project. And I was working on this. And the frustrating part is that I had already lost 2 years in another project that didn't work out. We ended up finding out that we were analyzing data. We were trying to reproduce data that was fake. So it wasn't true. It had been published, but we met the people that were working on this, and we finally understood that they had they had fraudulently, whether it was on purpose or not, the data wasn't true. So and I frustratingly, I had spent these 2 years wasting my postdoc time, and I was realizing that also all the pressure of the interaction between researchers, their PIs, and the academic environment, the bad part of the academic environment, which we can talk about if you want, but was very obvious at that time. And I thought, well, I like academia. I like science. I want to continue being involved in science, but this is not working. I don't want to become a PI running around grants and money and stressing all the time that I don't have enough publications. And it's just so stressful, where I just want to spend time thinking of problems and finding solutions to problems, right? Again, it seems a bit naive, but then this guy approached me in my former institute in Lisbon, in the Gulbenkian Institute. And he said, the person that's running the flow cytometry core is actually leaving in 3 weeks, and our director was asking me to make a list of people that I thought internally would be the best people to take over. And he knew that I was starting to look at alternative careers in science. I was thinking of communication. And then there's another factor here. I was a computational biologist. Although I loved my colleagues, we had amazing interactions, and we used to get along extremely well. But we worked in an office, right? We were just on our computers, and there was silence most of the day. The only times that the silence was broken was when I started messing around and or saying jokes or stuff like that. And they loved it. I know they loved it, but I was interrupting them, right? So sometimes I had to be careful and not exaggerate too much. But so one of the things I missed a lot was interacting with people. I need to interact with people. I've learned throughout my life that I'm an extrovert. I need interaction with people to gain energy, or else I'll start dropping. And that was missing, and I was realizing all these things at that time. And so the guy said, the first person I thought of was you, because you're a computational biologist, you love technology, but you're also into biology. There are a lot of computational biologists that come from physics or come from of informatics, etc., that don't really understand the biology, right? So I had the biology background. So I came actually a bit of the opposite. I came from biology, learned math, and then jumped into this field, which, by the way, wasn't called computational biology at the time. It was mathematical biology. Bioinformatics had just started. So it was all different. I know that people use these terms loosely as if they all mean the same, but it's not exactly the same. And so he told me that, well, you have all this background, so we think, and you also like people. And I remember him describing this, and I thought, but wait a minute, you're saying that there is a person that's sorting cells for other people? Like, what do you mean? I remember clearly imagining, but isn't it, I remember thinking of a Petri dish. I thought that people would, with you know how it is with in vitro fertilization, where you put a needle there. I thought it was a process like that. You put one cell over there, some of these cells, and that's how naive I thought about. So I didn't know anything about cell sorting or flow cytometry. I didn't know anything about core facilities, because remember, as a computational biologist, we don't really interact with core facilities. We have a computer. That was it. We ran stuff on the computer.
What year was this?
What?
What year was this?
What year? It was 2006.
Okay.
Probably summer of 2006, and I took over. I remember I started in November 2006, so almost at the end of the year.
Okay.
And so I knew Alexis because we knew each other. The institution was small. It had, I think, a maximum of 250 people, so 30 labs. So we knew all each other, and of course I knew Alexis. We used to hang out in the parties and, just or outside at lunch. I mean, you've been at the Gulbenkian Institute, right? You've seen the outside area where we have lunch. It was always...
Actually, I haven't. No.
Oh, you haven't? Oh, okay.
No.
I thought you had been there. They have to invite you. Yeah.
I don't think
I'll give a plug. But the thing was, when he started describing the concept of a core facility, so it wasn't, that's why I was saying I didn't choose flow cytometry. I guess flow cytometry chose me. But I chose the core facility, aspect. It could have been any technology. Actually, when I started learning flow cytometry, I had a huge help. When I think of this, I have to thank so many people, I'm so grateful for so many people throughout my life. I can't say I'm here in the position that I am if it weren't for so many people. And it's not like I'm just being nice and thankful. No, a lot of people helped me in the conversations that we had, the discussions that we had, trying to help me focus and realize what it was important for me. And this in particular, I remember thinking of the concept of core facility, the fact that you are involved with science, right? You're helping the researchers collaboratively, trying to figure out their problems, technical problems, but also related to the science. You have to translate the science to the technology, right? And be able to solve their problems. And I remember thinking, wow, I can become an expert in this, and this could be my thing. I could be really helping these researchers, and you get to interact with these people all the time. They come to you they handle. So it was a bit, for me, it was a bit of everything that I always wanted. It was the science, the interaction with people, the non-pressure of having to publish. I remember thinking, I don't have to publish anymore, and a huge weight just left my shoulders. And this was in the conversation of maybe half an hour, right? All this was going through my mind.
So, so for those listening that may be doing their postdoc or their PhDs at the moment, you say there's this weight lifted off, and they're thinking, yeah, but so what are you doing? Where's your impact to science?
Right.
But obviously there is it is an impact. And I bet you're still being co-authored on lots of publications.
Absolutely. That's the thing. And I have to say, but this is more of a detail, but I actually had many more collaborations when I was in the Gulbenkian in Lisbon, just because of the culture, right? People needed experts, right? And we weren't swimming in money. obviously. And so a lot of these things have to be, tinkered, tweaked. You needed to be very involved in the research that people were doing to be able to solve these problems. And I remember flow cytometry was just about immunology. We only served immunology, which was maybe 6, 7 of the 30 groups that were there. The nice thing at the Gulbenkian Institute, it was very broad. It had 4 main topics, but we had people studying plant biology, developmental biology, and of course using, so even evolution. Actually, the people who were studying evolution were working with bacteria. They were studying the mechanisms of evolution, etc. And they started using flow cytometry. So my objective, like from the first weeks, I decided my goal is going to be to bring flow cytometry to all of the institution. Because the first thing that PIs told me when I first started was, but why can't we use flow cytometry? Why is it only the immunologists? Why do you guys give priority to all the immunologists, etc.? And I said, but no, that at least my director told me that the objective of Flow Core is to serve everyone, equally. And so that's what I'm going to do. 5 years later, everyone was using flow cytometry in the Institute. And it was really exciting. And we had a lot of collaborations. I had probably so there were 30 labs and I had probably 20 ongoing collaborations with basically everyone. And it was really an exciting time where I learned a lot in flow. Again, like I said, I learned a lot from, Nuno Moreno. I learned a lot from him. Initially, I had a MoFlow. And that was my machine. There you go.
It's actually because you mentioned earlier tinkering.
Exactly.
And this was an instrument that needed
a lot of tinkering. Yeah. And I started with the MoFlow, and Alexis, he taught me everything he could in 2 weeks before he left, and how to work with the MoFlow, how to basically set it up and be able to sort. And of course, in 2 weeks, especially when you know nothing, by the time he left, he did a great job in 2 weeks. He did really teach me as much as you can in those 2 weeks. I don't know if you remember how he is, but he's, I don't know if he's ADHD or not, but he's an amazing guy. But when he gets excited, he's just, all over the place. And so I was trying to learn as much as possible, taking notes, seeing what he was doing at the same time. And so in those 2 weeks, of course, when he left, the first people that came with their samples, of course, lasers are not working. I don't have signal. Forward scatter. Maybe the forward scatter is not aligned. Maybe, there was so many things that could be wrong. And of course, I remember sweating, but keeping my calm, and then just telling the researcher, look, of course, you understand that I'm new here, but I'm going to make this work. So give me some time. I would go and call Nuno. Nuno would come in. I remember Nuno would just start taking apart the whole assembly, and I would go, oh my God, okay, we're not going to sort today for sure. But it was really exciting. It was just, I learned so much of the instrument, the technology, lasers. Nuno taught me to align lasers in the way, I can almost without a microscope, I can align pretty much any, from scratch. I remember with a fax scanner, Faxcalibur, changing lasers, the blue laser and just mounting it. And
so I have to ask, do you miss the MoFlow? Because it was the best teaching tool. And it sounds actually like this was a nightmare. And actually, I don't think the MoFlow was far from a nightmare. Yes, it needed a bit of a light. Once you had it singing, I know, personally, I felt at one. I felt the MoFlow was an extension of me when I was in that lab.
Absolutely.
It was part of you, and you could feel it, you could hear it, you could sense it. You knew exactly. It didn't take troubleshooting because you were part of it. And it was so... Wow.
And it would start working better when you were in the room, right? That's how, that's the relationship you establish with a machine like the MoFlow, right? It's almost like, I'm exaggerating, but the thing is, you can actually, as you're working with that instrument day by day, you're actually changing things in a way that somebody else that comes in will need some time to change it back to their way of doing things. I think the MoFlow, I miss it a lot. I think it's more of a R&D type of technology, where you could, for me, I did a lot of things with, like I said, we were the first to sort the sperm cell and the vegetative nuclei from Poland. This was the stuff. Alexis started doing this in collaboration with one of the labs, one of the plant biology labs, and they were trying to run this sugar solution as sheath, and it was a mess. I mean, imagine all the aerosols, and then as we were sorting, our hands on the table were, with the gloves, were getting, sticky because of all the sugar. So we had to clean it very well afterwards. But it was nice because as I went, as I started understanding the machine more and more, we started getting rid of all these, all of these different things that we would do, like changing the sheet. And we ended up actually being very successful doing it with just PBS, without even having to change the sheet. And it ended up, because I remember we were going to publish the method because we had all these modifications. But as I got better at it, I realized that no, we actually don't need any of these modifications. We just sorted just normally. So there was no really method to publish. But it was really exciting, the fact that we could change anything. We were sorting bacteria, yeast, even starfish, zebrafish, any mammal, dog, horse, mouse, human, human without the hood. Those were the days.
Yeah.
You could source.
I have the next question. So you took up the MoFlow, you started running a core, and this was back in Portugal. You're now over in New York. Mm-hmm. Alexis left, obviously, so you took over. Where did Alexis go first?
So Alexis went to Heidelberg to work with Andy Riddell at the time, EMBL. And then after Andy left, he took over. And then, they have this rotation. I don't know if it's 3 years.
Yeah, 9 years.
And then he left to Australia, where he is now.
Down in Melbourne.
Right. So yeah, I mean, and this is another thing of the field, it's not that big field, right? I think it's big enough you can always meet new people, but it's small enough that the people you meet, are still your friends. They're your colleagues, but they're friends also. And I that's one of the things I really like from this field. Again, I was saying that flow cytometry chose me when I started learning flow cytometry, especially with the MoFlow, with a microscopist beside me, and I would go and see their microscopes, and they would, explain to me all the things they were doing. I remember thinking that, this is a much cooler technology than, the MoFlow is exciting, but that it's moving towards, pressing buttons, but this requires not only understanding the technology, or let me put it this way, without understanding the technology, you're not going to help pretty much anybody, right? But with flow, I know a lot of people that really don't understand the technology. They read the user guide, press these buttons, we run the sample for you, and that's it, right? So in that respect, if I would choose a technology, I probably wouldn't have chosen flow cytometry, but I didn't know all these social aspects, right? The flow cytometry community is, I have the feeling that maybe it's not as spread as microscopy, right? Because microscopy is a general term. There are different fields in microscopy, right? Yeah. Of completely different.
I sit in both camps, and actually they're both really good communities, really good networks. And the microscopy community is very similar. The light microscopy community is actually really quite tight-knit, especially from the core side.
Mm-hmm. Yeah.
We all know each other really well. Just like with the flow cytometry, I would argue the microscopists dance slightly better at conferences than the flow cytometrists, but it's touch and go, and it depends on how much everyone's had to drink by that point.
Exactly. I should start going to those also.
Thinking of which, see, you sent me this picture as well, which is freakish. This is...
It is.
God, explain.
Yes. So this was, it happened, I think it was in Leipzig, if I'm not mistaken.
Isaac in Leipzig. That was the venue, the cavern downstairs.
Yeah, exactly. And I happened to have this, Microsoft phone, and it had an app that, that you could swap faces. And I just thought it was hilarious. I had done this already in my, back in my lab. And I thought, hey, I'm going to start swapping faces of all these people. And I mean, it's just hilarious when you see, you take the picture and then you wait a little bit as it's, swapping the face, and then suddenly beep, it appears and there the face is swapped. And it was just so funny. And so this in case, people can't recognize the two people here, I'm actually on the right with Rachel Walker's face, and Rachel is on the left with my face. And so this is what we were doing, swapping faces. From actually, I don't think I sent you. There's one with Paul Robinson and his postdoc that I swapped the faces. There's one with Rachel and Howard Shapiro, which I hold dear. Especially given the recent pass away of Howard, that was an absolutely remarkable, amazing human being, right? Not only, I think he was for sure a savant, he was just brilliant, a memory that I haven't met many people. Actually, I don't know if I've met people that have that capacity, that memory that he had, and you could discuss anything with him. But more than that, he was an amazing human being. Since he could remember everyone, so I can maybe tell you a little story about Howard. So of course everyone knows Howard. I knew Howard. I would see him in the meetings. People were always a bit dismissive with him because he came out sometimes very strange, or he was sometimes very abrupt because he was very driven, very excited. He wanted to know, okay, you are working on this. Why are you working on this? Have you noticed? And people would be really frightened that a person like of this stature would come up to them. And I remember in one conference, I forget which one it was, but very early on in my career, and he calls me out. He says, "Rui," and I thought, what? He knows my name? Holy shit! I go, okay. And I had my technician at the time, Telma, who is now in running. She's run several courses in Switzerland, and she was with me. And I come up to Howard with her, and I said, come with me. Let's meet Howard. And I the first thing I say, hi Howard. So this is Telma, and he goes, yes, I know who Telma is. And I thought, whoa, like, how does he know these things? So he must have gone to our website. He must have gone, done his homework or research. And apparently he needs to talk to me. Or maybe he just knows everyone. He's been in everyone's website, and which was pretty impressive. And I remember he started talking, about malaria. I forget now how he figured out, but we were, oh, right, exactly. Because we were collaborating with a lab in Lisbon, so not in my institute, but in the Institute of Molecular Medicine in Lisbon, where Paul Hutchinson used to be, by the way, used to work. And there was a researcher there that worked with malaria and had contacted Howard. And so Howard knew that we had a MoFlo, and so that we could work together. And so it was really, that was Howard. He knew everything. And at the time, I only knew him as, as the person that wrote the Bible of flow cytometry. But as we collaborated, I ended up going to Boston, working with him, with Tricia Rogers. At the time, she was at Harvard. Now she's at the Broad. And I remember we were collaborating, and we went to his house for dinner. And it was an amazing experience. Just being with Howard is an amazing experience. But having the pleasure to go there to his house, of course, he started showing everything, like the songs that he played, his albums, and he actually I forget. There's this duet, two guys, I don't know, from the 50s. Most likely you don't know. I forget now their name, but they were like comedians, but they sang. So they sang funny songs. And actually, one of the guys was on a wheelchair, and all of this was black and white. I had no idea who they were, and he showed me, and they had a song which was called Madeira wine, from the Madeira Islands, right, the Portuguese islands. He knew all about Fado, the Portuguese Fado. He knew a lot about Portuguese history, and I'm sure he knew about any other country's history. But it was just such. It was so amazing to see him so excited to show me, right? Why would he even bother, showing me these things? But he was like a kid. He was like a kid. Everything he did, he had the enthusiasm of a kid. And his humor was really, you could be offended if you wanted by his humor, but if you weren't offended, his humor was brilliant.
I think I know some people have been offended by his humor, but I think that's because they didn't know him. And if they knew him and where it was coming from, they'd understand actually this is on himself, not anything else. And he did that for a lot of people. Now, you mentioned the group and it was a start. You sent me this picture as well. Which,
Yes.
to be fair, looks like you are modeling to be part of a boy band, to be honest.
Exactly. So we were looking to take a picture. So we were just the four of us, and actually the person taking the picture was just kind of not an intern, but she was just there for a few months, and so we asked her to take a picture of us because we wanted a picture of the lab, and we said, okay, let's take pictures as if we were a boy band. And
so it was meant to look like a boy. Well, you succeeded quite well.
It was exactly. The funny thing is that this was actually the first picture we took, and I looked at my my friend Tiago that's on the left, right beside me in the yellow jumper, and I looked at the picture and I said, no, you don't look like we're supposed to be serious and tough, and no, you don't look tough at all. You need to you're almost smiling, so we need to redo the pictures. And then I don't know, I decided to boycott all the pictures after that. So I was just making stupid faces, although that one is pretty stupid already. But that one was on purpose. The other ones were just messing around with the pictures.
And you say messing about, you also sent me this picture.
Yeah.
Which is of you and Alfonso, obviously pulling the birdie above my head.
And then there's a guy there. So yeah, this was one of the nice things of being there in that institute was, so this is 5 minutes from where I worked, and so we would run courses, and this is a flow site course, by the way, the first flow site course that I was involved in and actually hosted it, and Alfonso and Tim Bushnell were the instructors, and we just it was just a lot of fun. We were in the class, and then we would go for lunch next to the beach. And this is just a picture, of us. Actually, this is in October, by the way. And this is how Portugal is in October, short sleeves.
Just rub it in. Yeah.
Right. Still some people in the beach, as you can see, not many, but, it's October, right?
Yeah, but the classroom's just the same, isn't it? It doesn't matter what the weather is outside. Inside.
It doesn't matter. Actually, so this picture is from Zagreb. This is also a flow site course. So that first course that we ran, I believe I'm going to say 2011. That was in the beach, and then I started. So with Alfonso and Tim, I started lecturing for flow site and also Excite. So that was more or less at the time where Excite started. And I remember I was running a lab. I hired one and then two technicians, and at least every month I would go to and give one of these courses in some place in Europe. And I remember Tim saying, we'll give you a stipend, so we'll reward you financially for this. And I kept telling him, no, just pay the expenses, pay me the trip. Because just the fact that I'm going to other labs is just, for me, is such a rich opportunity to see how people do things, how they work, how they run their labs, etc., their core facilities. And so for me, it was an opportunity actually to learn, and at the same time I could teach. And teaching is also a learning process. It also teaches you a lot.
And this is Dennis.
And that is Dennis Polachek. Yes, and we had a lot of fun. I went twice to Zagreb. Actually, I was going to continue going there every year, but then I moved to the US, which was a pity because we also a lot of fun there in Zagreb.
I wanted to ask. We've only got about 15 minutes left, and I wanted to ask. That's a big move going to the US from Portugal. How did you find it? With family at the time?
Yes.
So I presume they were supportive. There must have been a lot of trepidation about moving.
Yeah, I mean,
how did you find it?
So the thing is, when I was doing my PhD, the idea of my PhD, so this was back in 1998. when I started my PhD. So I finished my degree in '97. I was 6 months working in a lab, and then I started my PhD right after. And the idea was to stay 6 months in the US, learning all these tools, these computational biology tools, and then I would go back to Portugal and work in the lab. So both in the lab, but also both in the wet lab and in the dry lab. Building a model, testing that model, going back and refining the model, and to do these things back and forth, which I thought was the way to do, the best way to do science, was having that structure, that, as I said, formalizing things mathematically, so you can, predict, test, and then go back and refine your model. And so I went to the US. I went to the University of Michigan. I fell in love with not only with the culture, the lifestyle, but also with I was in the University of Michigan. So that's where they announced I forget his name, where they were doing the genome sequencing, that Craig Venture, I believe.
Yeah.
That's when he announced, it was there in a lecture, that they weren't going to spend 10 years or whatever it was. He said that, I remember it was like the end of the year, and he said next May or next April, we'll have the human genome sequence, and everybody was blown away. So, you're in the middle of all these things happening, and I thought, I'm going to go back to Portugal? Why? Like this is it. This is where I want to be. And so at the time, I got married 5 days before I left to the US, because, I thought I was already predicting, and my wife kept telling this was the least romantic way of asking to marry someone, because I remember we were thinking, I'm going to the US, but what if I want to stay? What if we want to stay there? If we're not married, it's going to be very difficult for you to move, right? So if I try to justify a visa for my girlfriend, and so one day I just turned to her and said, "Hey, I'm going to go down to the, whatever you call it, the office, right? And I'll get the paperwork for us to get married." And that was my marriage proposal. And
how many children do you have?
And now two.
God, you're so lucky that she didn't just throw you out at that point.
Exactly.
It's just unbelievably lucky.
So then, I married 5 days before I left. And, I left to the US 6 months later. I said, no, we need to stay here. We need to. So she flew over and she spent like a week or two. She thought actually at the time she was a bit hesitant, but then we had the opportunity for her to actually do a degree here in the US. And so she ended up coming. We spent another 3.5 years, I think a total of 4 years in Michigan. And it was one of the best times in my life. We were young, we were just enjoying life, and science was great. And then, unfortunately, since I didn't finish my PhD by the time my fellowship ended, I didn't have a visa anymore. So I had to make that decision of, whether I throw away my PhD and restart again after 4 years, or I go back to Portugal, finish it, and try to come back to the US. And that's what we did, except it took us 13 years to come back to the US. But it was always in the back of our minds, both my wife and I. And actually, we had our son, he was 8 months old when we moved back to Portugal. So we already had the son. Very difficult times. We didn't have money. We didn't have a job. I didn't have a lab to stay. Again, I was helped by incredible people that took me in their lab. They just allowed me to work. Here's a computer to finish your PhD. Maybe you can work for us, in some projects. And that's how I actually started my postdoc afterwards. And things just got better for both of us. And and then by the time I moved to flow cytometry, I managed to gain a reputation in such a way that one day I started looking out for jobs in the US, and I had this opportunity to come and work at Sloan Kettering, and I took it without hesitation.
Love the way you say you gained yourself a reputation. That can be a good or a bad thing. So question, quick fire questions. Portugal or US?
US.
Michigan or New York?
New York.
Oh, You're on safe ground. Mac or PC?
Now Mac.
Now Mac. Okay.
I've changed throughout. I've been a Mac guy. I changed to PC guy. Now I'm a Mac guy again. I'm actually agnostic.
No, beer or wine?
Wine.
Red or white?
Absolutely. That's no doubt.
American or Portuguese wine?
Portuguese.
Ah,
I love Portuguese red wine.
my favorite at the moment is a Portuguese red. I have
Which one?
I've about 36 in the garage stocked up, along with other bits. But
Okay.
a good batch, you got to enjoy it.
Exactly.
Coffee or tea?
Tea.
Do you drink coffee?
I drink coffee. I mostly drink coffee, but I enjoy more tea.
Okay, just imagining what you're like on a lot of coffee. Chocolate or cheese?
Cheese.
Ooh,
No doubt.
okay. Early bird or night owl?
Early bird. Well, actually, both.
Especially at
something.
conferences.
Yeah, that's a balance I've been trying to get. But yeah, both actually.
Okay. What's your pet hate? What do you not like?
As a what, pet?
No, as in a pet hate. So something that annoys you.
Oh, something that annoys me. Oh,
Don't say podcasts.
right. Oh, I don't know. It's a difficult, I haven't thought about that. I tend to try to be very non-judgmental. So I really enjoy pretty much everything. But of course, there are things that annoy me. Let me see. I don't know. I hate when people waste my time. I really don't like that.
Okay.
They don't respect that that you have a life, let's say.
And we shall quickly move on. What's one of your passions in life outside of work?
Outside of work, I would say people. People are my passion. I just love, I think about traveling and things like that. But what I really like of traveling is meeting different people, and so even locally, I just, I could go to a bar as long as I'm with the right company. For me, that's, we can be outside, not even in a bar, anywhere. As long as I have good company, that's what I love best. People.
Do you have any bad habits?
Oh, yes, I do. Like staying up late and waking up too early. That's what I'm.
You must have more bad habits than that.
Oh, yeah, I'm sure. I can't think of any at the moment.
It's always dodgy when you won't confess to it.
It is.
What about sports or hobbies? What do you do in your?
Yes, I play soccer, or actually, I should say football. I'm already very Americanized here.
Right, soccer's good.
I play football. Actually, I'm just recovering from this pinky finger here. Yep. Last Sunday. So I was playing as a goalie. So I play a lot as a goalie because I
trained it back.
Right, and it came out and it went out of the socket and towards the inside. So I thought I had broken it.
Dislocated.
But no, it was just dislocated. So now it's feeling really. But yeah, that's what I enjoy. I play every week. I used to play 2-3 times. Now I'm playing maybe once a week, or I try to.
So if you're a soccer fan, Ronaldo or Fernandes?
Ronaldo.
Who's the better player today?
Today, yeah, that's questionable. But Ronaldo still has the experience, right? So
I'm a Man U fan, so I love both. so that's not such a big problem, is it? Right. TV or book?
What?
TV or book?
Oh, book. Absolutely. I'm only starting to watch TV now.
What sort of book? Fact, fiction?
I try both, because I usually tend to go to fact. I like a lot of, historical books or books about politics, science. But then it gets too serious and I need to relax a little bit. So I always, I have a reading. Sometimes I'm reading both, at the same time, or I'll just alternate between a fact book and then fiction, because I really enjoy fiction also. Any type of fiction.
What's your favorite movie?
Ooh, that's a good question. My favorite movie, I think it would be. There are a lot of favorite movies. It's always difficult for me to say, but I would say probably Once Upon a Time in America is probably up there.
Okay.
for the movie, but also the music. Yeah.
So we have got 5 minutes, and I've got a few questions. So I'm going to have to cut down and just think about which question best to ask. I think we've heard about your inspirations.
Mhm.
I think Alexis obviously played a very big part in that very early on. And you're right, he does go off on tangents quite frequently. Would you say taking that job, would you say your undergraduate, the start of, was probably the most challenging time of your career so far?
The undergraduate, no, I wouldn't say. I mean, it was very challenging, right? And I was mostly disappointed. So it was more challenging in that sense that, okay, I have to finish it and get over with because I'm not learning anything. So like the last 2 years were really tough because I just wanted to move on and start working in the lab. But no, I would say the most challenging time of my career was when I went back to Portugal because, like I said, we didn't have any money, so it was more both a personal, but also I didn't have a lab to be in. I didn't have support from my supervisors, and so a lot of my PhD I did it by myself. I had huge help from informally from a colleague of mine that supervised me, Fernando. He was amazing. He took all his time to help me, finish my PhD. So he was really an important person in my life at the time. But it was really challenging because, we didn't have any perspectives. Things were looking really, really dim at the time. And we just said, well, we can't go backwards because there's no way, to go backwards. So we just have to move forward. And so it was a time, it took maybe 3-4 years where we were underwater. And I remember once in a while feeling like I could breathe a little bit and then go underwater again and just wait and things will get better. And they did. And now I look back and those were amazing times, times that I learned a lot about life and about people, about our profession and all these things. And so it really taught me a lot. But I think those were the more challenging, even more than when I came here to New York to run this huge facility, even more than that. And I think that helped a lot. That experience helped a lot.
With my one final, not so serious question. If you could have any job in the world, what job? What would you do?
Oh, shoot. I think I've thought about that, but I don't know. I really enjoy what I do, and I've been thinking a lot about, is running a core facility what I do. Actually, I've had the opportunity to become, director of cores, and I don't even, that's not something I like, so I like people a lot. Managing people, that's a different thing. I like people when I don't have to manage them.
Yeah,
right. It's a completely
I took that step, but I kept my cool.
Right. So, I really like what I do. I like solving problems, right? So any job where I can, so I'm not, fixed in science and flow cytometry. At the moment, I could change to something completely different. And it would be, exciting to learn something completely different. And I think, that's one of my favorite characteristics is that, I know that I'll be, hopefully, if I'm 90 years old, I'll still be excited to change, to move, to leave my comfort zone. Leaving your comfort zone is one of the most difficult things, but it's also one of the most gratifying things when you start becoming successful. It's really nice. So
finally, on a far less serious note, you said earlier when you were a computer scientist, not calling that back then, but computer scientist, that actually everything was quite quiet until you started talking and telling jokes and stuff. So what is your favorite joke that's clean?
Oh, shoot, I don't know. Yeah, no, I've never. So I love dad jokes. I can say that. So maybe I don't have a joke in particular that would be my favorite, but definitely dad jokes, which of course my children, always scoff, when I tell them these jokes. And actually, most people will scoff when you say a dad joke.
Example. Go on.
I don't know. There was one just yesterday. Yeah, like at the top of my head, if you put me in a spot, I don't know. That's why I'm actually not a comedian, because if I'm put in a spot, I forget everything. But if we're having an informal conversation, the jokes will start coming out.
I thought this was informal. Come on.
Yeah, it was informal, but like when you're put in the spot, I don't know. They're especially jokes like Larson, the comic strip, right? It's this kind of humor I love. I can say I've been influenced by a lot of great comedians like Benny Hill.
Oh,
A lot of people would question whether that would be a comedian. But I used to watch Benny Hill so much when I was young, so much. And it was now I look at it and I go, oh, that was so
I asked your favorite joke, not a confession.
right. No, it's yeah, and I don't know. I'm trying to think of other comedians that inspired me, but yeah, it's difficult to to think of a joke.
Okay, so one final question.
I know I'll think of it when we turn off the Zoom call.
We'll just keep recording, and we can put it as an outtake at the end of it. How's that? So who would you like to see next on Flow Stars?
Oh, that's a good point. Who would I like to see? Well, I would definitely like to see Howard. I think Howard would have been an amazing person. I don't know. There are a lot of people I would think of. I don't know from the top of my head. People here in the U.S., for instance, Joanne Lanigan. I think she's had an amazing. Oh, one person I would love to see is Tricia Rogers. I think people should know. She runs the core at the Broad Institute. She's pretty known here in the U.S. I don't know how well known she is in Europe and other places, but I think she, as a person and where she is and knowing her background, how she got there, she's an extremely inspiring person, and I think everyone should know her. How do you say her path?
Yeah,
yes. I think if I would think of somebody to interview, it would be her, definitely.
Thank you, Rui. That will go on the list. Rui, thank you very much for joining me today. Everyone who's been watching or listening, thank you for joining us. You've heard about Alfonso, Tim Bushnell. You've heard Derek Davis dropped into this. You'll see a lot of these people actually on the other podcasts in the past, and you've just heard of who I've got to go and invite next. Rui, thank you very much for joining us today.
Thank you. It was a pleasure, Peter. It was a pleasure. Thank you very much.